So welcome from my side to everybody attending this week's environmental ergonomics session. It's a pleasure again to welcome you all. And as you know from the advertisements, today's session will focus on cold physiology and in particular on brown adipose tissue metabolism, thermoregulation, etc. Before we go to the session, I would just like to share a few general housekeeping informations for you to better enjoy the session. In particular, those of you who have attended our previous sessions, you know of our now infamous Wall of Fame. So this is the Wall of Fame of all the presenters from season 1. And then this one, I think the next one is the Wall of Fame of season 2. We already had a lot of excellent sessions on different aspects of environmental physiology, science, communication, etc. And you know that you can find all of these sessions on our webpage, icee2021.com/past events. These seminars are co-organized by Stephen Chung from Brock University, Chris Tyler, who is today also with us from Northampton University, and myself from University of Ljubljana. It's also worth mentioning that for all of the sessions that we had that were presented, we also the speakers also present short student handouts, which can also be found on our webpage under the names of the presenters, which can be used for teaching purposes, etc., etc. So it's really a nice educational material to use in your courses as instructors, lecturers, etc. Also, a relatively new initiative was started by Stephen, I think a month ago. In particular, he started setting up short educational videos discussing recent interesting publications from the environmental physiology or ergonomics area. And these short videos are also available on Environmental Ergonomics YouTube channel. But you can also, but the purpose of them is not, is for to use for the general public, so to better, to improve our outreach of our science or science that we do in the field to the public, to everybody. So it's really a nice way if you have time. You definitely should have a look at them and also disseminate through your friends, family, and colleagues. Before we go to today's session, I would just like to remind you that in 2 weeks' time, so on April 14th, we'll have the final session of this season 2 of the environmental ergonomics series. The topic will be social thermoregulation in humans with 2 speakers, and it's also worth mentioning that we will be celebrating 1 year's anniversary on that day, so you should get prepared for some surprises. So hopefully we'll see you all there. So not to be too long, now we will move to today's session. As I mentioned before, we will focus mostly on brown adipose tissue, and for that we have 2 excellent speakers today. One, Wouter van Marken Lichtenbelt, is already with us, and I believe that Hitoshi will be joining us really soon. The first one will be presented, the first talk by Wouter, on the key players on cold and metabolic health between brown adipose tissue and or skeletal muscle. And I will now stop sharing my screen and will give the floor to Wouter, please.
So I'm unmuted now. Can you hear me?
Okay. Yeah.
Yes. Okay. Thank you very much. And thank you very much for giving me this opportunity to give this presentation and for the nice communication between you. Okay, I see Hitoshi is there as well. so the second talk will be given as well. Okay, so I'll start then. Yeah, oh, let's see. Sorry. Yes, so we got interested in the thermophysiology for a long time already, and we saw there was more and more opportunity to use temperature as a novel lifestyle factor. Of course, we know nutrition and we know exercise, and there's coming more and more factors that appear to be very important and can be implemented in daily life to increase our metabolic health. And with temperature, we see many beneficial effects of both heat and cold. We study them both, but now, of course, I will focus on the cold. So let's jump into that. And I will present some older studies just to give it the storyline, and also some brand new studies will be presented. Yeah, to start with this, of course, you know that in the cold, you increase your energy metabolism, and you do this mainly by shivering, and you have a tremendous increase. So in this study from Haman, we see there's a heat production increase of more than 3 times as much heat production in the cold compared to thermoneutral. And that can even be 5 times higher, which is tremendous and which really makes the body work. And that's one of the things we think about, is that if the body has to do some work, it's good for your metabolism, it's good for your health. And but already some time ago, we found out that before the shivering starts, and here you see in the lower graph, you see the EMG indication for shivering. But before that starts, you see there is already some non-shivering thermogenesis. So there is an increase in energy expenditure without any muscle contraction. And that is interesting. So we thought, let's continue in that field, follow up the older studies of Dancy. And so there were some older studies, but not that many. So we really took a larger group of 20 people and looked at what happens in the cold. And you see on the group level, which is the top graph, you see there is indeed a significant increase in energy expenditure. And interestingly, and I'll come to the acclimation studies later, we see there's this increase in the same subjects in winter is higher than in summer. On the lower graph, you see there's quite a lot of individual variation, some up to 30% and some really close to 0. So there is a huge individual variation, sorry, in this thermogenesis. And there was one, the Iceman, I'll come to 1 or 2 slides later in my talk, who even went to 40% of resting metabolic rate without actually shivering. So it can be significant, that's clear. But the individual variations, we have got interested in that, and also in group differences. So here we study in the respiration chambers in Maastricht, and we had people for 48 hours in the chambers in a thermoneutral and in mild cold conditions. And we see that there's a difference between the lean subjects and obese. The obese have a blunted response, and the lean subjects clearly show the significant increase in the cold environment. And finally, as an introductory slide, I'll show you here some of the studies from Kingma, who studied energy expenditure in elderly. And here you see that the younger adults clearly show what I showed before in the earlier slides, an increase in energy expenditures. That's the open circles on the left graph. But if you look at the elderly, you see there's even a decrease in energy expenditure, probably because they cool down and maybe a Q10 effect. And you see this has also its effect on the body core temperature, as you see on the right side. So yes, there is an individual variation in non-shivering thermogenesis, and there's also group differences. And then the next step was which tissue is responsible. And we have been looking in skeletal muscle a lot in the past, but this talk is about brown adipose tissue. Let's dive into that. And brown adipose tissue is, of course, well known from different animal species, especially the smaller animals that also live in cold environments. They need in winter times, especially, they need brown fat. But it's also known, of course, from young adults, from babies. There's a significant amount of brown fat. So all that was clearly known in the past, and we know that brown fat really distinguishes from white adipose tissue. Brown fat has much more mitochondria in which the heat production takes place. It still has some storage in the form of fat droplets, and it's highly innervated by the sympathetic nervous system and has a huge perfusion. So it's really different from white adipose tissue that are really fat cells that are filled up with fat, really for fat storage only. Brown fat is really the stove of the body. It can be turned on and produces heat. And that heat is produced by a special uncoupling protein. At least that is responsible for this heat production. So as I told you, the sympathetic nervous system activates the brown adipose tissue, and I'm not going to talk into all these details that you see here, but it is still interesting to note that once the cell is activated and a cascade of responses occur and reactions, normally in the mitochondria, with the electron transport chain, ATP is formed. You're all aware of that. But in the case of brown adipose tissue, there's UCP1, the uncoupling protein 1, that more or less shortcuts the proton gradient over in the membrane. And that means that the energy normally used for ATP production is now not used. It's by the shortcut, and then heat is produced. So heat is released. There's still, of course, some ATP production, otherwise the cell wouldn't survive, but most of it turns into heat. So this UCP1 is really characteristic of brown adipose tissue. And of course, the cell needs to be fueled. That's why it's highly perfused, the tissue, and fatty acids and glucose can take up. So this is a general overview, already an older picture, because especially from mice and rodent studies, a lot was already known for some time. And you see these acute effects, mainly this UCP1 activity, and you see chronic effects with acclimation. You see there's more UCP1, more mitochondria, etc., etc. From the same animal studies, it was also known that brown fat was related to metabolic health, not only an increase in energy expenditure, which may be advantageous for weight maintenance or maybe even weight loss, but also that it uses glucose and lipids, so it could be used as a glucose or lipid sink in the body, and also that there's links to insulin sensitivity, so it may improve glucose metabolism as well. So it was a nice surprise that it's already more than 10 years ago that we and some other groups found out with the onset of modern imaging techniques that also adult humans have brown adipose tissue. So we could make this visible with using the PET-CT scan, and the PET-CT scan is from nuclear medicine technique. And the most important, or at least the one that is most widely used, is the tracer FDG. So it's a fluoride glucose molecule. And there's no time to explain you all the details, but you have to realize that all the pictures that we always see are from glucose uptake. So there are some other traces, and there's a lot in development, but this is where most of the data comes from. So it is glucose uptake and not all of the metabolic activity of brown adipose tissue that we actually map. Nevertheless, it's a very interesting option to show brown fat, as you saw in the pictures, and also that, that this is also giving you functional information. So here you clearly see that if we see this huge individual variation in non-shivering thermogenesis, I lumped here data from several studies, and there is a significant relation with this brown adipose tissue glucose uptake. So yes, brown fat seems to be involved in the non-shivering thermogenesis, also in adult humans. And now I come back to this group differences. We also see a clear relation with the body composition. So obese subjects have no detectable or much less brown adipose tissue than lean subjects. So yes, there's a relation with body composition, and several groups also show there's a relation with age. So that comes up to what I showed you at the start with the data on non-shivering thermogenesis that was also lower in the older and lower in obese subjects. But now the question arises, what is the actual contribution of brown adipose tissue? And this, I must admit, has not been solved yet. So we are still posed with the problem, how much is active is brown fat? There have been several estimations, as you can see here, but there are also already studies from quite a few years ago, and there's not much news going on, although there are many different new techniques and new traces that in near future, I expect we will get more insight into what the actual contribution of brown adipose tissue is. But the estimates go up to 5%, and maybe there are some individual values that we have shown up to 60%, but it's really exceptions. So in general, you may say, yeah, it's about around 5%, 2 to 5%, something like that. The future will learn how much it actually is. But as you have seen, that the non-shivering thermogenesis itself can go up to 30%. There's more going on than just brown adipose tissue, most likely. Now I have to give a few slides on what happens when you increase brown fat. You saw from the animal data that brown fat can increase, that there is a acclimation effect, but it's good to know that there are different types of brown fat. And in the past year, there have been lots of developments and new insights in the types of cells. And interestingly, that during embryological development, and these processes have become more clear that brown adipose tissue is more closely linked to skeletal muscle tissue than to white adipose tissue. They are different lineages. So that is very interesting to see, but you do see on the right side in this picture, you also see the so-called recruitable brown adipose tissue. We also call them beige adipocytes. And they are more closely linked to white adipose tissue, but have the same characteristic to a large extent as brown adipose tissue has. I think I'll skip this one. So there is so much interesting research has been going on, and this one also clearly shows that there can be trans differentiation in these brown or beige adipocytes, and especially in the lower part of this graph, you see there may be precursor cells in white adipose tissue that can, in case of cold or in case of beta-adrenergic stimulation, be turned into brown-like adipocytes. But when you are back into thermal neutrality, that can be formed into white adipocytes again. So there is a lot of dynamics going on, and there seems to be the possibility of trans differentiation. And so it's interesting to note that in humans, most of the brown adipose tissue is in fact bright adipose tissue. I think that most researchers are now agree that we have a lot of bright-like, brown-like adipocytes in our body. So we do have some deeper layers with classical brown, but most of it is beige or bright adipocytes. This picture is a little bit too simple. It's more complicated, but it gives you just some overview on the different types of fat we have in our body. For us thermophysiologists, the most important is that also the beige cells can be turned on for heat production. So functionally, that's the most interesting. So we have seen now that there's a relation between non-shivering thermogenesis and body mass index and age. We have seen that also brown adipose tissue is related to that. So then the next question that we raised is this really linked to the cellular characteristics of the brown fats. And therefore we did a study in cells. So we could take biopsy material in a lot of patients that were undergoing deep neck surgery, and we isolated the stromal fraction, and we could be able to raise a cell culture. And then we measured cellular respiration in a seahorse, a special device, but in fact it is just measuring oxygen consumption, and we could stimulate the cells with norepinephrine, mimicking the sympathetic nervous system. So here you see where we got the tissue. Yeah, okay, just as an illustration, we really got the samples. I hope you're convinced. And then here you see the expression of UCP1, which is characteristic of brown fat, as I told you, and leptin is a hormone that's characteristic for white adipose tissue. And you clearly can see we took both white and brown adipose tissue, and you clearly can see that we got the right tissue under study. This graph shows the experiments we did. So we have a state 4, in which means it's the uncoupled state. And if you, with the uncoupled state, measure the oxygen consumption, and you then activate the cells by norepinephrine, which is indicated by NE, you see there's in brown adipose tissue, there's an increase in energy expenditure, way much more than the increase in the white adipocytes. So yes, it's functionally active also in culture. And then we looked at the group that we studied, and we see that this oxygen consumption upon NA, upon norepinephrine stimulation, you see that is also negatively related to body mass index, and not so in white adipose tissue. It's only in the left graph with the brown. Next graph here, you see the same is the case with age. There's again a negative relation with oxygen consumption in brown adipocytes with age, and not so in the white adipocytes. So this shows there's the same trend that we also see on the whole body level. And we see this, finally we see here that UCP1 is itself, so the protein itself is related in the same way to body mass index and age. So all in all, we may conclude that the mitochondrial function in adipocytes from brown adipose tissue negatively relates to body mass index and age, and that you may use the UCP1 really as a predictive measure for brown adipocyte activity. But is now the message this bad? I mean, can't we do something about it? Can people who are obese, who also often have the metabolic syndrome, maybe insulin insensitivity, so maybe having diabetes, can we do something about it? In other words, can brown fat be recruited, like we know from animals? So that's in fact one of the first studies that we did, bigger studies that we did after we discovered the brown adipose tissue. So that was a study with 10-day cold acclimation. And we hope to see the same as we see here on the left with this picture from rodent, from Chinti. There we know in animals it can increase tremendously the amount of brown fat. So people were kept for 6 hours per day at 14, 15 degrees Celsius. There may have occurred some shivering, some tense muscle. Of course, during the measurement of the brown adipose tissue itself, there was no shivering. That was really done in mild cold situation. And here you see one subject, you see a clear increase in brown adipose tissue. So yes, it can be recruited. And we measured, I think, 17 subjects, and 14 out of 17 showed this increase. And parallel to that, we again show an increase in non-shivering thermogenesis. So cold acclimation has an effect. So the next step we did was we studied people with type 2 diabetes, because we also wanted to study in more depth whether it also has an effect not only on energy expenditure, but also on insulin sensitivity. And here you see that we did, in fact, same kind of experiment, 10-day cold acclimation, the same temperature. So it was really a repetition, but now we include also a clamp a hyperinsulinic euglycemic clamp to study insulin sensitivity, and we took muscle biopsies. So here are the results. We see in these groups, we see there's an increase in energy expenditure in the mild cold after the cold acclimation, after this 10 days. That's the left graph. You see there's a slight increase in brown adipose tissue activity, but here I must say, maybe the PET-CT scan using a fluoride glucose, a glucose tracer, is not the best tracer to use because the tissue can be insulin insensitive. There may be really differences in glucose metabolism. So glucose tracer may not be the best tracer. Nevertheless, we see a slight increase and a significant increase in brown adipose tissue, but it's still not much. We were very much surprised to see that the insulin sensitivity, nevertheless, was tremendously improved, more than 40% increase on average, which is comparable to the best treatments there are. So cold, yes, has a very positive effect on insulin sensitivity. And we saw it's not significant, it's not in the publication, but I just looked at the data later again. But there is a trend towards this change in insulin sensitivity and the increase in brown adipose tissue. It's a trend, maybe it's a power issue, maybe it's also because we haven't really used the right tracer to study brown adipose tissue in these patients. Interesting though, if you look at the increase in glucose uptake, which is here indicated by the SUV mean, sorry for that, but it's just a measure, but it shows how much glucose has been taken up. Of course, you see the significant increase in brown fat, as I showed you, but also this is SM, the skeletal muscle. You see there's a significant increase in glucose uptake, showing that skeletal muscle was more active. So we studied in the biopsy material in more detail this tissue, and we saw that there was really an increase in blood 4 intensity, especially in the membrane, sarcolemma membrane. So we see really there's a translocation of the GLUT4, the transport of the glucose in the skeletal muscle. So the conclusion then is it's not only brown fat that plays a role in the improvement in insulin sensitivity, and maybe it's the skeletal muscle is even more important. So we're having a puzzle here. We do see a very functional brown adipose tissue, but we see that it's maybe not working on its own. And now I go back to an older study, just 1 or 2 slides of there where we studied the Iceman, in which we saw that this man had really a huge non-shivering thermogenesis capacity. So the Iceman is very famous, Wim Hof, you probably all know him. He's having his training program to make you more resilient and feel better, etc. But we studied him in our lab several years ago, and there we saw that he really had a huge, as you see in the left graph, top graph, a high non-shivering thermogenesis capacity. But what we also saw is that his brown fat was not as high as we expected. We thought that he would have had tremendous amounts of brown fat, but he didn't. And we also saw that the respiratory muscles took up a lot of the glucose tracer. So in fact, the respiratory muscle and brown fat both may be responsible for this increase in the heat production. So again, it shows there's muscle involvement as well. He also does his Tuma meditation things, and that's a special way of breathing. So he's breathing in a special way. So that's why maybe this respiratory muscle activity is higher than we saw in other subjects. So in conclusion, we may say that brown fat and skeletal muscle may be teammates. So they work in concert. So yes, cold acclimation leads to metabolic improvements, and both skeletal muscle and brown fat may work together. And this leads us to think of a kind of temperature training, like you have exercise training maybe cold acclimation, you may call it a temperature training. There are a lot of things happening in parallel, improving the metabolic health, although the mechanistics, the cellular mechanisms may be different, but there's also a lot in parallel. But then how cold do you actually need to be? And we recently did a study, just finished and published, on cold acclimation, where we thought, well, let's see that we really, during the cold acclimation, prevent any shivering at all. So that was, if someone even started a little bit of shivering, then we warmed the subject up again. So there's really no shivering occurring at all. But that means the temperature is higher, of course. We have now 16 to 17 degrees Celsius. We did a meal test, we did the clamp study again, and we took muscle biopsy. And I'm not going to explain, there's no time, the whole study, please read it if you want. But we saw some improvements. I'm not even going to talk about that. But as you can see here, during the meal tests, we see no difference in plasma glucose. We see no difference in triglycerides, although we had expected in the cold to have, after cold acclimation, to have maybe lower plasma glycerides as well. And if we go to the insulin sensitivity, which is on the right graph here, we see there's no significant improvement in insulin sensitivity, not immediately after the cold acclimation, and also not in the long term. So what can we now conclude from all this? There are a lot of things that I mentioned, but one of the things is that some people say, well, this brown fat is so little, it's just a rudimentary tissue. Well, I don't think so. I think it's really responding so functionally, so the amount of brown fat should be important. So it's probably really a functional tissue, maybe also contribute to a maybe lesser extent our energy metabolism and direct effect on insulin and glucose and triglycerides, maybe not that much, but there's more and more data and lots of interesting studies on brown adipose tissue as a secretory tissue. So I think this may be a very important issue, that brown adipose tissue is regulating metabolic responses, or in concert with other tissues, regulating it elsewhere in the body. And these are just some examples. There are coming more and more factors that are important and interact with other tissues. There's a very nice overview here, this where I said crosstalk from Schele and Wolfram. They have a nice overview last year. Please read it if you're interested. And they also highlight the close vicinity, how you say that in English, the close togetherness, being together, the nervous system and the brown fat location is always close to where the nerves are. So maybe there's an interaction between the nervous system and the brown adipose tissue, even more close than we think of. Maybe there can even be afferent neural signals from brown fat to the central nervous system. And there's also nice studies that is Klingenspoor and others, and the Finland group, they are involved in that they show from the meal secretin release, and that may directly affect brown fat, and that may influence then satiety in the brain. So there are very interesting links, and then maybe the brown fat comes into picture again in our improvement of metabolic health. So yes, brown fat is functional responding. It is related to obesity and age, and as also has been shown in animal studies. It is, and that's very interesting, recruitable, and also in obese and type 2 diabetes. So you can improve the amount, increase the amount of brown fat. The actual contribution to the energy metabolism is not that high, as far as we know, but better imaging techniques are needed. But very interestingly, brown fat is also a secretory organ, so it may be related to other organs. So it's really working together with other tissues. And that is, I think, the future of brown fat. And we should look at that and try to understand why cold acclimation has so much healthy effects. And we're studying how much muscle activity is actually needed for improvement of the insulin sensitivity. So it's really, I think the muscle again is also in the center of attention. And with that, I would like to end this talk. I'm really happy to answer some questions.
Yes, thank you very much, Wouter, for an excellent presentation. Lots of data, you know, lots of studies included, but really insightful into the main areas that you and your group cover. Maybe I just have a question in regards to the cold acclimatization interventions that you mostly used, like a 10-day intervention, for example, you know, and check the effects, so pre and post effects. What do you think about, or what do you know about the effects of prolonged exposure to cold? For example, if you had a group of individuals and then you would expose, you know, half of them to a prolonged cold environment and then the other ones to a relatively warm environment, would there be any functional difference in regards to the brown adipose tissue and stuff like that in terms of the prolonged effects?
Yes, that's a good question, and it's really interesting to study that, because the scanning, the PET-CT scanning is invasive because there's radiation involved. You cannot do that many because normally you would like to measure during the cold acclimation at several time points and see whether the amount of brown fat levels off, or whether after 10 days it's still increasing but could increase even more. So that is in fact not really known. I think these long-term effects are very interesting, and it's generally so with this metabolic health improvements, and especially in mild cold. And we are also thinking of having cooler environments, just daily environments, and see whether you can improve our health by that. That are really long-term effects and extremely difficult to study. So that it's a very interesting question, but we don't know the actual really long-term effects. What we did see after the 10-day cold acclimation, the first study we did in type 2 diabetes, that there was still an improvement in the glucose metabolism even after two more weeks. Yeah.
Okay, thank you very much. So it's really interesting. And is there any difference between, for example, populations that maybe live in a cold environment, like in the, I don't know, in the north, and then, you know, people living in hot climates? I mean, like on a populational level?
Well, we do know that also in Africa there are people living there still have brown adipose tissue. There are some indication that there's some lower levels of, but I think Hitoshi will tell about that, in Asian people. But I don't know whether there are really people exposed daily in the cold. We know of the very classical study from Heaton, that they show that outdoor workers really have a lot of brown adipose tissue, relatively much. Yes.
Okay.
Yes.
Yeah, thanks a lot. One could imagine that, you know, given the potent effect of the cold, that it should have an effect if you're habitually exposed to it. And now, before we move on, there's a question from Takayuki, and I believe that Chris will be able to enable her to ask a question herself. Chris, can we manage that? Or yeah, Takayuki, you just need to unmute yourself. Okay, apparently we don't have a question here, so maybe we'll leave that for the general discussion after Hitoshi's talk. And then, Wouter, if you can just unshare your screen, then we'll move on. And as I mentioned before, our next speaker is Hitoshi Wakabayashi from Japan. Hitoshi, thank you very much for joining. And now, generally, the floor is yours. You can share your screen, and we'll discuss your presentation after the talk. Excellent.
All right. Can you see the slide?
Yeah, we see everything. Thank you. Yeah, you can start.
Sorry for my late joining this virtual environmental ergonomics. So I misunderstood 1 hour later. So let me talk about that. So thank you for inviting me to talk in this event. And let me talk on the variation in human blood for tissue activity and conducive thermogenesis. So again, I'm Hitoshi Wakabayashi, working in Hokkaido University, Japan. Right. Let me briefly introduce my university. This is Japan, and Tokyo is here, and my university is located in the city of Sapporo. And this is Hokkaido Island, the northern island in Japan, and we have a very cool climate. And my department is here in the center of the campus. So it's only 5 minutes walk from my home to my campus, but it takes another 15 minutes to my office. So, and in this summer, we have a big event, and Sapporo City welcomed the Tokyo 2020 Olympic Marathon. And the marathon course crossed our university for 3 times in the race. And my office could be one of the best place to see the event. But I'm not sure I can go into my office during the event. Then this is the pictures in my university, many flowers and trees, natures. And yeah, this is actually the Tokyo 2020 marathon course. So the condition of the road surface is best ever. And in winter, we have a lot of snow like this, and we cannot run the marathon course anymore. And we do some activity like the tug of war on the snow. And anyway, this is one of the best place to study cold environment. Okay, this is the overview of my presentation today. And I briefly explained the human blood pulse tissue. And I will talk about the inter-individual variation in blood pulse tissue, like population difference. And I will talk about the coordination between the non-shivering and shivering thermogenesis. And then I will talk about the intra-individual variation in blood pulse tissue, like seasonal variation and circadian variations. As Wouter also presented, in 2009, several research groups in the world, including Professor Saito in my university, found the activation of blood pulse tissue in human adult. The left figure is the PET image in mild cold and thermoneutral environment. So you can see the lot of glucose was uptaken into the supraclavicular and paraspinal regions in mild cold condition, but not in the thermoneutral condition. So this result clarify the cold-induced activation in blood pulse tissue in human adult. Additionally, as shown in the right figure, they shows significant correlation between the human BAT activity and their body mass index. So considering this result, blood pulse tissue would be the key organ for thermogenesis and also improving the metabolic health. So that's why many researchers from a lot of research fields like health science, nutrition, and thermal physiology focused on this tissue. And cold-induced thermogenesis, CIT, could be classified into the sibling thermogenesis and some non-sibling thermogenesis. And this is the model of the whole body thermogenesis, including resting metabolism and non-sibling thermogenesis and sibling thermogenesis as a function of the cold stress. And we also show the thermal effectors of each metabolic components. And in thermoneutral condition, only the resting metabolism is activated. Then in mild cold environment, non-sibling thermogenesis is induced. And then in colder environment, sibling was onset. So as Wouter has presented, the contribution of the skeletal muscle and adipose tissue on the non-sibling thermogenesis is one of the interesting topics. And also the coordination between the sibling and non-sibling thermogenesis could be the another interesting point. I will explain later. Okay, let me talk about the inter-individual variation in blood adipose tissue activity. Back and colleagues compared the blood adipose tissue activity in South Asian participants and Caucasian participants. As shown in this PET image and also in the right figure, Caucasian participants showed the greater amount of blood adipose tissue volume compared to the South Asian participants. So since they born in the same country and lived in the same climate zone, so there could be some effect of the genetic variation on the activity of human blood adipose tissue. Then another group focused on the variation in the uncoupling protein 1, the UCP1, that is located in the inner membrane of the mitochondria in the BAT and played a role for regulating the thermogenesis. And they focused on the non-sibling thermogenesis during mild cold exposure in different UCP1 haplotype, that is genetic types. So, and they found that this haplotype GMGTAN showed the greater increase in their oxygen consumption during the mild cold exposure compared to the other haplotype groups. Then they focused on this haplotype, and based on the 1,000 genotype database, they analyzed it like this. This figure shows the correlation between this haplotype frequency in several populations and the annual temperature where they live. And you can see, for example, the European group who live in the cooler environment show the higher frequency of this haplotype. But compared to that, African group or South Asian group who live in the hotter environment showed low frequency of this haplotype. And East Asian shows a quite big variation. So from this result, there could be effect of the long-term cold acclimatization to the haplotype of the UCP1. Then a question came out, if we have a less amount of blood adipose tissue, there should be another thermoregulatory response to regulate our body temperature. And there could be the other thermal effectors for offsetting the less BAT activity in individuals who has less amount of BAT. This is the study conducted by Blondin and his colleague. And they focused on the contribution of blood adipose tissue and skeletal muscle. And left figure shows the net glucose uptake per unit tissue volume during cold exposure. And as you can see, the BAT shows the highest glucose uptake compared to the other organs, these skeletal muscle or subcutaneous fat. However, considering the large amount of the skeletal muscle, the total glucose uptake into the skeletal muscle is far greater than that in blood adipose tissue. So because of the larger amount of skeletal muscle, they suggested the skeletal muscle would be the major sum effector for cold-induced thermogenesis. Additionally, they focused on the type 2 diabetes patients who has a less amount of blood adipose tissue. And the left figure shows the net glucose uptake to subcutaneous fat, skeletal muscle, and blood adipose tissue. And type 2 diabetes patients shows less glucose uptake into the blood adipose tissue compared to the healthy young control. Whereas the glucose uptake into the skeletal muscle is significantly greater in type 2 diabetes compared to the healthy young control. And also, they evaluated the shivering intensity using electromyogram, and they showed type 2 diabetes patients tend to show the higher shivering intensity compared to the young controls. So considering these results, shivering in skeletal muscle might offset the less blood adipose tissue activity in the individual who has a less amount of BAT. According to these previous report, we hypothesized that humans with less blood adipose tissue would have more contribution from skeletal muscle to non-shivering thermogenesis and or shivering thermogenesis. And we investigated the contribution from each organ to non-shivering thermogenesis and shivering onset during gradual cold exposure. So let me explain our study. That is the protocol for assessing the BAT activity using FDG PET-CT. 18 Japanese healthy males participated in this study, and they stayed in 90 degrees Celsius mild cold environment for 2 hours, and they got FDG injection at 60 minutes. And after 2 hours cold exposure, they scanned by PET-CT. And BAT activity was evaluated by standard uptake value of FDG, and the detectable BAT volume where SEV higher than 2.0 was evaluated at cervical, supraclavicular, and axillary regions. And based on this analysis, we separate the participants into high BAT group and low BAT group. So this is the physiological characteristics of each group, and they don't show any difference in percent body fat or skeletal muscle mass, but the higher SEV mean in high group and the higher BAT volume in high BAT group. Then they conducted this gradual cold exposure test. Starting from the thermoneutral baseline, we gradually decreased the temperature to 18.6 degrees Celsius, and they stayed in the mild cold environment for 90 minutes to induce the non-shivering thermogenesis. Then we decreased the temperature to 12 degrees Celsius in 30 minutes for initiating the minimal shivering. And we measured the metabolic heat production, core skin temperature, and electromyogram on the pectoralis measure for assessing the shivering intensity and shivering onset time. This is the result of time course change in metabolic rate and the shivering intensity during the gradual cold exposure test. During the mild cold exposure in 18 degrees Celsius, high BAT group showed the high metabolic rate compared to the low BAT group. And both group doesn't show the significant shivering during the phase. Then during the shivering initiation phase, low BAT group showed earlier onset and greater intensity of shivering. And both groups shows a similar metabolic rate during this phase. This result suggests the blood adipose tissue contribute non-shivering thermogenesis, and shivering thermogenesis might offset the less non-shivering thermogenesis in individuals with less amount of blood adipose tissue. And we also focus on the multi-organ contribution to non-shivering thermogenesis. This figure is the correlation between BAT volume, skeletal muscle mass, and the resting metabolic rate and non-shivering thermogenesis. You can see the significant correlation with the BAT volume and non-shivering thermogenesis, but not with the resting metabolic rate. Whereas skeletal muscle mass showed strong correlation with the resting metabolic rate, but they don't show the correlation with the non-shivering thermogenesis. So from this data, we suggest blood adipose tissue contributes non-shivering thermogenesis, but skeletal muscle shows a minor contribution to the non-shivering thermogenesis. However, as we explained in our manuscript, there is some limitation in the estimation of the skeletal muscle mass in our study. And also, we only focused on the volume of each organ and the correlation with the whole body metabolic rate. So we need some further investigation with evaluation of the tissue-specific metabolism. And this is one of the original finding in our study. Left figure shows the BAT volume is positively correlated with the rectal temperature at the end of the mild cold exposure phase. And this rectal temperature is positively correlated with the shivering onset time. So these results suggested individual with less blood adipose tissue activity shows a less non-shivering thermogenesis, and it results in the lower core body temperature. And lower core body temperature induces the earlier shivering onset. So it could be the indirect coordination between some effectors for offsetting less BAT activity. So this is a summary about the inter-individual variation in BAT. Individual and population difference in blood adipose tissue activity and UCP1 haplotype was observed. And individual with more or less blood adipose tissue induced more or less non-shivering thermogenesis. And from our data, the skeletal muscle mass did not correlate with non-shivering thermogenesis, but it needs further investigation. And shivering thermogenesis could offset the less non-shivering thermogenesis in the individuals who has less amount of blood adipose tissue. Then let me move on to the next topics about the intra-individual variation. And this picture shows the seasonal variation in the poplar trees in my campus. and human BAT activity. Professor Saito in 2009 already reported the seasonal variation in the brown adipose tissue activity. So it is higher in winter compared to the summer. So there could be seasonal acclimatization in human BAT activity. And Yoneshiro and colleague investigated the seasonal variation in non-shivering thermogenesis and body temperature regulation. The left figure shows the cold-induced thermogenesis in high BAT and low BAT group in summer and winter. And especially in high BAT group showed higher non-shivering thermogenesis in winter compared to summer. And the right figure shows that change in tympanic temperature during the mild cold exposure. And again, the high BAT group showed less reduction of core body temperature in winter compared to the summer condition. So this result also suggested seasonal acclimatization in brown adipose tissue activity, and it could affect the non-shivering thermogenesis and body temperature regulation. And recently, my research colleague, Dr. Matsushita, investigated the circadian variation in BAT activity and cold-induced thermogenesis. This figure shows the cold-induced thermogenesis in mild cold environment in the morning and evening in low and high BAT group. So they found that high BAT group showed higher cold-induced thermogenesis compared to the low BAT group in the morning, but at the evening, no group difference was observed. And also, cold-induced thermogenesis tend to be higher in the morning compared to the evening in high BAT group. So these data suggested the circadian variation in BAT activity and cold-induced thermogenesis. We additionally analyzed the data using the IR camera technique. We used the temperature difference between supraclavicular and chest region as a surrogate parameter of the brown adipose tissue activity. And right figure is the showing the temperature difference in the morning and evening in some neutral and mild cold environment in low and high BAT group. And the high BAT group shows the greater temperature difference. That means greater activity of brown adipose tissue compared to the low BAT group in the morning and evening, but there is a difference between the morning and evening that is the high BAT group shows a greater temperature difference in the morning compared to the evening. That means they show the higher BAT thermogenesis in the morning. So this result also supports the evidence of the higher brown adipose tissue activity in the morning. Additionally, they also focused on the circadian variation in diet-induced thermogenesis, DIT. This figure shows diet-induced thermogenesis relative to the energy intake after the same amount of breakfast and lunch and dinner in low and high BAT group. And they found the high BAT group showed greater DIT compared to the low BAT group after breakfast and after lunch, but not after dinner. So it suggested BAT contribute diet-induced thermogenesis after breakfast and lunch, more than that after dinner. It's also suggested there could be a higher BAT activity in the morning. This is the summary about the intra-individual variation in BAT. There is a seasonal variation in BAT and non-shivering thermogenesis that is higher in winter than in summer. And circadian variation in BAT and non-shivering thermogenesis was also observed. That is higher in the morning than evening. Okay, thank you for your attention. They are the research colleague and graduate students support and contribute my work. And thanks again, Teddy and Chris and Stephen for giving the opportunity to have a talk today. Thank you very much.
Yes, thanks a lot, Hitoshi, for your great talk. Again, I heard that Hokkaido has really great winters. I know that skiing should be nice, yeah, based on the snow that you get there. So it's definitely, I have to agree that it's a great place to study cold physiology in a way, yeah, during winters. So I have a short question in regards to the study that you presented that was published in EJAB. In particular, was it how did you find individuals with higher brown adipose tissue content and lower? Was it difficult to recruit, or because there were big differences between the two groups? Yeah. So how did you do that?
About the recruitment of the participants.
Yes.
So we normally ask the student or colleagues around us, and they participated in our study. And then at first we tested the PET-CT protocol for assessing the BAT activity. And then we separate the participants into the two different groups. So,
okay.
Yeah,
very nice. So, and the other thing that I was wondering is, you mentioned that there is a big individual variability in the, on the one hand, on the amount of the brown adipose tissue that people have, but also on the responses to the cold exposure. Yeah. So is there, because we know I come from hypoxia area, and there's a really large individual variability to responses. Do you maybe know if somebody is exposed to cold, is the response of brown adipose tissue repeatable if you do it 3 times in a row? Does it happen, is it always the same?
So starting from the final question, the repeatability of the BAT activity. So actually we didn't do repeated measurement of the BAT activity. So because of the some effect of the radiation. So normally we do the just one time assessment of the PET-CT protocol. Then we we separated them into the two groups, considering the activity of brown adipose tissue, and we tested the metabolic response of them, and we discuss the mechanism, or we check whether the brown adipose tissue activity affect on the physiological response or not.
Okay, thanks a lot. And now we have a question from Koichi, and he's wondering if the higher brown adipose tissue activity in the morning compared to the evening, is it possible that can be explained by an overnight fast?
Okay, thank you for your question. We control the amount of the food before the each protocol. So I think that's why there's no effect of the fasting throughout the night. I normally, I forgot the exact time, but we set, for example, the 10 hour before the experiment, we set the time and the amount of the food before each test in the morning or evening.
Oh, okay. So the food intake before the actual test is standardized so that you exclude the effect of dietary differences. Okay, that's great. Now I just have, I think, a final one from my side, and I think this is for both of you. Potentially, I was wondering through the talk, because you both mentioned that in obesity, individuals have much lower brown adipose tissue levels or, or activity compared to lean individuals. And is this due to the fact that obese individuals have lower, actually lower levels of brown adipose tissue, or just the activity is so much lower?
I could answer that one.
Yeah, thank you. Yeah.
Sorry. Yes, that's of course a chicken and egg question, but I think that they really have a lower brown adipose tissue because we have a personalized cooling protocol. So we cool them up to shivering and then increase temperature so that we really have mild cold, non-shivering conditions. And then we do the cold exposure for an hour, and then we do the PET-CT scan. So that means that the lower amount of brown adipose tissue that we find in obese is really, that's really the case. It's not a cooling thing. The other question is then, why do they have less brown adipose tissue?
Exactly. Yeah.
And that is the chicken and egg question. So is it because they have already better protection against cold environments? And have less brown adipose tissue because of that, or the other way around. I mean, I think if we see how well a brown fat can be recruited, that is the case. So they have less because they are less cold, because they have better protection by means of their body shape, by means of the volume to surface ratio. They have less skin compared to their body mass, and by means of their subcutaneous fat layer. So the same cold environment isn't experienced as cold in the obese.
Yeah, okay, so that sounds like a really reasonable answer. So Chris, do you maybe have any questions from your side, or?
No, you just asked the ones I had written down.
Okay. So then I see that there are no other questions also from.
I could answer one more about the brown fat coefficient of variation. Of course, we, as Hitoshi clearly explained,
of course,
we cannot do repetitive measurements, but we did it with the non-shivering thermogenesis. And then we had the summer-winter comparison. And although the non-shivering thermogenesis was higher in winter because of acclimation, there was a clear relation between the subjects measured in summer and winter, meaning that those that have a higher non-shivering thermogenesis in summer also had in winter. So there is, with respect to non-shivering thermogenesis, a clear relation, and that means if we also know there's a relation between brown fat and non-shivering thermogenesis, that could mean that indeed, if you do repetition in brown fat, that the ones with high would have high again in the second measurement. Sorry, my answer is a bit, is it clear?
No, it sounds reasonable. So it would be repeatable if you do it, if you would repeat it, probably it's the same. And then maybe since I just have it's more of a subjective question, but how difficult it is for participants to endure the cold adaptation, cold exposures? Is it for them subjectively really difficult, or is it easy, or how do they respond to that? I've never done a cold acclimatization protocol, so I'm not sure, how difficult it is for them.
Shall I? Hitoshi, you want to answer, or shall I?
Yeah, can I just answer quickly? Yeah, actually the participants in our study, yeah, feel quite uncomfortable during the protocol. Even in the mild cold exposure, they felt quite uncomfortable to the cold environment. So probably repeated cold exposure as a strategy to improve our metabolic health needs some yeah, idea to much more comfortable condition. But yeah, I think the cold stimulus is the best way to activate the blood for tissue activity. So
yeah, I can add to that. That's true indeed. When you start the cold exposure, they are uncomfortable, but during the 10-day cold acclimation, the uncomfort rises to just comfortable or just uncomfortable. So it gets more comfortable during the acclimation process. So you get used to it. And this is what we know also from general other physiological experiments, that you really get used to it physiologically, but also psychologically.
Yeah, I guess habituation plays a big role there because it's tricky, and we know that most of us don't really feel well when we're cold. So, and for implementation of such a protocol in in a population level, it's. But I think on the other hand, probably this uncomfortability is an important part of getting there. Yeah.
Yeah. But you get used to it. So yeah.
There's no way around it, I guess, to make it work. Yeah.
Yeah.
Okay, so if there are no other questions, then I would really again like to thank both Wouter and Hitoshi for their excellent contributions, and again just remind all the participants that in 2 weeks' time we'll have obviously big celebrations of 1-year anniversary, and social thermoregulation topic with Hans Isermann and Olivier Duyol, both from University of Grenoble. And I sincerely hope we see you all there. Keep well and keep healthy, and hope to see you again very soon. Yeah, goodbye.
Okay, thanks again. Bye-bye.
Yeah, thank you, Wouter. Yeah, bye. Thanks.
Thank you very much.
Yeah, Hitoshi, thanks a lot. Yeah, for your help. Yeah, sorry.